So what is molecular diagnostics? Any thoughts on this? Molecular. I mean, you know that a protein can also be on a molecular scale, or a peptide. But when people talk about molecular diagnostics, they usually refer to nucleic acid. Could be DNA, could be RNA, could be both, could be something which has nothing to do with genomic information, but with something which is circulating. There's also homework on this one. Or like this diagnostic test, what we just heard about, CRISPR-Cas kind of test would also be a new type of molecular diagnostics. Now, let's get to the introduction. Where does this all come from? The reason why molecular diagnostics is so popular is simply because it's easy. Why? You have 4 nucleotides, and you just need to understand in what kind of a sequence those 4 nucleotides pop up. The mechanism behind it is super conservative. So throughout all the species, basically the way how to replicate information is always the same. There's 0 difference between a bacteria all the way up to a mammalian cell, with a few exceptions, but the concept is always the same, and so on and so forth. And then it's also a very simple biopolymer, and that also led to very easy resolving conditions, so that, you know, Nobel Prizes were given to Crick and Watson, and probably they were the worst Nobel Prize winners ever, because Mrs. Franklin and others did all the work, as you know. Nevertheless, it took off, and there were a few selected cases where it became really, really interesting. That is, of course, the introduction of Sanger sequencing. Sanger sequencing, Mr. Sanger, he also received a Nobel Prize to really sequence basically nucleic acid for something where I don't even know what the sequence looks like. This is basically the foundation for all of our knowledge of what we currently have in the databases, all the way until next generation sequencing supported the throughput, but that still is the gold standard as of today, no change since the 70s basically. And then obviously PCR showed up at some point in time, and PCR basically, when it was introduced, in particular quantitative PCR, everyone said, you know, now we solve the problems of the world. Okay. It did not look like this, but what PCR really helped us, I mean, significantly, was how to treat infectious diseases. Because here we are just looking for usually small genomes, you know, a virus or anything like this. And molecular diagnostics is just outperforming anything else. It has the highest sensitivity, specificity, and therefore also high predictivity values. So that makes simply sense. But for anything beyond infectious diseases, as you're going to see also in the examples, was not so great. And then sequencing popped up. And then again, the people said, yeah, we're going to heal the world, right? And now, 20 years later, we realize, yeah, maybe not. It's just another tool, another element of supporting, and what is the focus of our discussion, the differential diagnosis, what we're having. So most of the stuff you know anyway, so I'm going straight into some use cases. And kind of related to the discussion what we had today on having some cell-free DNA. This is one of the hot topics that we currently have. Why is this? Because blood is what? It is the best systemic source of any kind of information related to the body, because not only nutrition, oxygen is being transported, but also garbage. And garbage may also has some information in it, and that could be, for example, cell-free DNA. And the cell-free DNA is basically something which comes in any cases, because when cells die, and they continuously die in your body, the components of your cells go into the blood stream, and then they are usually chopped up, you know, smaller pieces of these things here. These are the histones, these are the proteins where the chromosomal information is wrapped around it. Yeah, so a highly positively charged protein. And then you have the negatively charged DNA. Keep in mind, we're working under physiological conditions. Weak acids, weak bases are always charged. Yeah? So charge makes quite a bit in this setting, what we are looking at. And then the people have to simply separate the DNA from the protein structure, and then they do the analysis in what we see then later on how this actually works. And what do they hope to see? Well, they want to see not only that, you know, any kind of a dying cells, but what they hope to see is, do we see also tumor cells. So it's one of these things where we are getting now into this overdiagnosis regime. How much of dying tumor cells can we actually see? Why does this make sense that we can see this? Because tumors have an aggressive growth. Usually vascularization, what we just had a chat also in the other presentation, cannot uphold, meaning that many tumor cells are simply dying because there's no oxygen left, right? So the periphery is growing faster than the vascularization can keep up. And that means you always have a signature of tumor cells in your bloodstream in form of circulating cell-free DNA or ctDNA or whatever they call it. Yeah, there are all kinds of acronyms on this one. And this would be actually interesting for diagnostics. And the key question, and that's your homework, how could one recognize the origin of this circulating cell-free tumor DNA? I mean, just measuring that something is there doesn't mean that you know where it is, right? But if you would then take someone who had a very early screening test, a simple blood test, this is the hope what we have, then you would also like to know, where should I do the imaging? To just make sure, where is it maybe? Any thoughts or anyone has an idea how we can get organ-specific information? Maybe you find additional protein information, for example, which is expressed specifically in a respective organ. Yes, could be. Or maybe there are also other things. And one of those things which we are going to look at here in this case for your homework is epigenetics. What is epigenetics? So there's a different level of regulatory mechanisms on top of DNA. And that is that our cells do methylation of certain nucleotides. And when those methylation take place, the body can switch off certain genes, literally, because then our enzyme machinery recognizes this as, ooh, I shouldn't touch it, right? And this is super organ specific because our genetic makeup is all the same from head to toe. There's no difference, but still we have different organs. And the different organs only being generated by switching off or more off or less off by these epigenetic markers. This is this methylation pattern. Yeah? There's also another consequence of this methylation pattern. The body recognizes what is the older sequence from when they do a replication, for example, from the newer sequence. So if there is an error, the body actually recognizes is the error on the fresh sequence, which is not methylated, and does the repair on the right side, and does not introduce a new error in the hard copy, essentially. Okay, so those are the things how the body regulates.
Brings my question to my mind. Like, don't we have different proteins for each epigenetic?
Yes, we do.
How we understand which organs it comes from?
It needs to be somewhat correlated. And there is an opportunity to correlate if you find, for example, this kind of cell-free tumor DNA in so-called small vesicles or exosomes. And these exosomes also carry proteins, because an exosome is like a mini vesicle that is being squeezed out of a cell. And sometimes they also carry nuclear information and protein information. And with this, you could get a very holistic pattern about where this exosome comes from. So that's the next step beyond, beyond this level, what we are going to talk in a second. Okay, but we're going to get to this point anyways. So what is GRAIL now doing? GRAIL is a kind of company sponsored by the largest sequencing company what we currently have, is Illumina. And their goal is to develop an in vitro diagnostic test by looking essentially into this cell-free DNA. They do all kinds of isolations, library preparation, which we're going to talk later on, maybe even amplification, and then they do targeted sequencing. But what they're doing, they sequence also for the methylation pattern. And so when they sequence for the methylation pattern, they do know where this kind of a cell-free DNA was coming from, what kind of an organ. Okay. And now there were a few studies going on about, oops, I've hidden this already. That this may be the success story. And what I would like you to do is, for your homework, have a look if this is really a success story. So the claim was a 1-year acceleration of access to the gallery test for the US population has the potential to save 10,000 lives over a 9-year period. Think about if this is a success story in cancer, okay? And think about what do they actually see with this test? And is this biomarker really pushing the limits? And think about it from the perspective of molecular diagnostics is an easy thing to do. It's not so complex, not so difficult as doing protein analysis or more complex tissue analysis and so on and so forth. So this would be your homework for next time. Now, let's skip a few years and let's go a little bit back in history. When I was a student, no, I was not a student anymore. There was this nice publication in 2001. The covers of both Nature and Science showed basically that the human genome was solved. And in the one case, in the Nature paper, it was the government-backed consortia using Sanger sequencing, so the gold standard to resolve the genetic makeup in the different chromosomes that they had. And Science pushed something called shotgun sequencing, basically using a stochastic principle to align snippets of sequence information into what can come back again to a whole chromosome. And we're going to look into this, as I said, but to make you a little bit of aware that hope, hype, and academic fame, here we've solved the human genome. But if you have a look in 2001, we only saw 2/3 of the genome. 1/3 was completely masked, and only last year, actually, we're getting close to a complete sequencing of a genome, and that's only for selected people, meaning that this was difficult to sequence. And why was it difficult? Because a lot of our genetic information is very repetitive, meaning I don't know how many repetitive units I have from point A to B. Was the position of A to B very short, or was it really long because of repetitive units? This was one of the major problems, for example, what they had. Or stretches where the nucleotides were all the same. Yeah, was it now 20 A's or was it just 15 A's? Those were the difficulties what we had. And by combining different technologies, which are capable to provide long stretch information and highly resolved short stretch information, only now we can really claim that we are getting almost close after 20 years of this claim to a whole genome. Now that's a very famous figure. And since we are mostly electrical engineers, I have to raise my voice here. People said, sequencing has been outperforming even Moore's law. So what is Moore's law? Remember that? Who is an electrical engineer? He has to disclose himself now. What is Moore's law?
Exponential.
Exactly. So every 18 months, we double the amount of field effect transistors what we're having. Yeah, more or less. Let's just put it this way, okay? So Moore's law, I mean, he was the founder of Intel, right? He came up with this experimental law, and this would be this line here. Now, what is Moore's law based on? What technology is behind Moore's law? What was the driving force? Mainly. The transistors, yeah, but how do you make the transistors? By which process? Lithography, exactly. So it was one kind of a technology which kind of scaled down over the decades and allowed us to make smaller and smaller size sheets. That's it. Sequencing is improving and generating throughput by switching from one platform technology to the other. So you cannot compare those two. Yeah, this would be Apfel and Birnen to the power of 2. Just doesn't make sense. And as you can see, everything up to here is more or less driven by Sanger sequencing. And then, so on the y-axis, this is the log scale for the cost for a human genome. And then we had this introduction of shotgun sequencing, or what is called now next generation sequencing. This one is now this exponential drop. And now with things where you don't have to spend too much time on the sample preparation, you're getting now close to the situation that you can really sequence a whole genome for $1,000. I mean, this is really impressive, right? Isn't it? Yeah, it is. But what do you do with this information? And do you have sufficient computational power to work on it? Do we have the databases to make some kind of a sense out of it? And that's exactly the difference between the life science claim and the clinical reality. Just because I know a mutation, and what is a mutation? What is the norm human? Yeah? Is there a norm human without a defect? Do we have anything like this? So you get the point. Yeah? So from a clinical perspective, that's very, very different from this perspective of what I can achieve just because I have the technology in my hands. Now going back to CMOS, because I love it and I've worked in this field for a few years at IBM, this is the 3 nanometer node. I mean, when I was a student, it was super cool to have something like less than 130 nanometers. Oh. Now we have a 3 nanometer. It's crazy. And just to give you the size reference, an antibody has a length of 10 nanometer. Okay, so the gate regime, this is always the smallest feature size of the field effect transistor. Yeah, this is the node, the 3 nanometer node. That is just the obnoxious part of it. Question to you: do you know the key limitations of CMOS? Ever thought about this? Everyone is talking about the end of Moore's law, the end of CMOS, but why is that? Yes. Yes, so you have the so-called short channel effect. That's the biggest issue. So this is the driving force why you have to improve your gate almost exponentially to the linear decrease of size. So that's the biggest challenge what we currently have. And if you can see on to these pictures here, this would be a wire with a wraparound gate. So that's currently basically the state of the art. FinFETs, wraparound gates. Those are the transistors how we are working on. What are the other issues? I mean, how many silicon atoms do you have in a cubicle of 3 nanometers side length? Exactly, you can even count the number of silicon atoms. How does the doping work under these length scales? Ever thought about this? I mean, how do we dope? Yeah, we are adding dopants onto our silicon material in a more or less stochastic manner, right? On average, yes, same density. But if you scale down, the variance per unit volume becomes dramatic. So the concept of doping does not work anymore. Okay? And then the third issue, and that's actually even the bigger issue, is simply heat. You're generating now so much heat that you can, make your eggs for breakfast very easily. And at some point in time, the whole system would just collapse because there's too much temperature being generated per unit volume. And that's also the reason, so when I was at IBM, the big thing was how to cool down the kernel. So we drilled holes into the kernel and pumped liquids into it, because a liquid has much more heat capacitance, and therefore you can take more heat away. And that's the way how big servers are working these days. They're not in air anymore. The high performance servers are dumped into oil or in water. Otherwise, they would not work anymore. And so on and so forth. So those are the limitations from an anorganic perspective, but this is all top down. Okay? It's a deterministic device, what we're looking at. But what we are having here is not deterministic. I mean, it's deterministic from the perspective of we have a double chain structure, But anything around this, the whole machinery which is being used for molecular diagnostics, is not deterministic. It is a biological machinery which we adopt to please our needs for technical solutions. And that's something what I would like to guide you over the next few minutes, step by step, so that you recognize, okay, now the schematics, what I see is actually all wrong. The reality is always gray, and I have to always optimize my biological conditions to run sequencing, PCR, and so on and so forth, because there are limitations due to the chemistry involved, which is required for performing molecular recognition, sequencing, or polymerase chain reaction. Now, PCR was the core of this idea, and this was really ingenious. And that guy here, this is Kerry Mullis. He was working for a small company in California. He's a surfing biochemist, my hero, basically, as you have heard it just a minute ago. When he gives interviews, he usually is heavily stoned. But nevertheless, he had this ingenious idea that if I want to do amplification, I need to have a second primer. One primer will not do the job. And his idea went into a patent. Roche bought a patent, a single patent, for 100 million. This was, in those days, an obnoxious amount of sum. Would you say this was a good deal or a bad deal at those days? What's your guess? I mean, Roche was one of the most successful pharma companies in those days. 100 million for a single patent. Please. So why would you make a business deal like this? What would you like to gain? Yes. Yeah. Perfectly fine, but actually I give a shit about this. I'm only interested in income, in profit margins. Yeah. So how long did it take Roche to have an income of more than they spent on that single patent? That was the key question. How much was it? What's your guess? How many years? 3 years. They were laughing in the cellar a lot when they paid the company, being super happy. Oh, we got 100 million from Roche. And Roche said, you have no idea what you're doing. Okay. We had a similar case in the TOM, for example. Remember when we talked a little bit about functional imaging with positron emission tomography? Was I discussing? Maybe a different lecture. Okay. Point is, you can get specific information where the tumor is by, having an antibody which targets a certain part of your organ where the tumor is sitting, and there is a positron emitting a label on it. And this was developed basically all the way to the final clinical study stage here at TOM with other partners. And then TOM sold it for a few millions to a startup company. And that startup company sold it a few weeks later for $1 billion to a pharma company. I'm not kidding. This really happened, right? And it's one of these typical German success stories that you understand. Academia is academia. Okay. So in either case, having said that, what is amplification of DNA? It is not a deterministic kind of a process, but we customize basically the biology for solving this technical task of amplifying whatever is here. And for this, we're basically adding, the basic components, the nucleotides. We're adding the polymerase. So this is the enzyme taking care of the amplification step itself. And this is Kerry Mullis' only work. We're adding the 2 primers for determining where actually this amplification step should take place. But most of the infectious diseases, what we're actually having, are RNA viruses. SARS-CoV-2 is an RNA virus. Does PCR work on RNA viruses? No, it does not. We need DNA. RNA would not work. So on top of that, for making this useful also for infectious disease diagnostics, the people had to add more of the biology, like this reverse transcriptase. It's another enzyme. And what it does, it just helps you to get from the RNA level to the DNA level. And then from the DNA level, you actually start to do the PCR reaction. Okay. So, it's not that simple. The reality is actually much more complex if it really has to become of clinical relevance. Now, let's go through the details. What is eventually PCR? Check. And we're going to go back again to this question, why is one primer insufficient for the whole PCR reaction? What you're doing is, at the end of the day, let's assume you start with DNA, you're happy and you have a double strand, or you have your RNA strand, even a single RNA strand. You have to first get this into a so-called complementary DNA sequence. But at the end of the day, the PCR reaction always starts with a piece of DNA as a double strand. Why is this a double strand? Because DNA is usually only stable in a double strand, or likes to be in a double strand conformation. And what is now important is to understand, as any biopolymer what we're having, all biopolymers are polar with respect to their ends. Yeah, an amino acid biopolymer has a carbonic acid and an amino group on both different ends, right? This is also polar ends. And the same is also true for your sequence of nucleotides. You have a so-called 3' end and a 5' end. Now we're going to look into the details what that means again. And of course, the inverted strand with the exactly inverted sequence of nucleotides is the 3' to the 5' end. We have our desoxynucleotide triphosphates, so our reagents carrying already the energy. As you remember, triphosphates, ATP, stuff like this. This is always energy carrying molecules. We have primers. The primers are very short sequences which guide us, like CRISPR-Cas, basically where to do the amplification. And then we have the obvious polymerase. Now, to get access to the hydrogen bonding area, of course, we have to get rid of this double strand bond. And for this, we do a denaturation stage. Denaturation means I usually have to heat up my system. We talk about that one bucket. So everything literally happens to occur in a bucket like this. Okay. We first denaturate so that we have access to the nucleotides and the hydrogen bonding area. And then we have an annealing step. The annealing step happens to occur at a different temperature. Under these conditions, the small primers can diffuse around and find a sequence, which is just a match to their own sequence. Then they bind. And because we have two primers, we have one for the first strand and then one for the second strand. That's a key element for the amplification process. And the third is then the extension, meaning when the primer has bound, then the polymerase will sit at the very edge and starts to go from the so-called 5' to the 3' direction, or as I keep telling it also to my kids at home, it's always better to have a 3 in an exam than a 5, and that gives you maybe the guidance to remember what is 5' and 3'. And this happens not only on the one strand, but this happens also on the second strand. And if you do so, you end up with what? How many strands do we have now? After this first round? 4 strands, okay? The 2 original ones and the first 2 copies. And now we start all over again. The original strands, they are kind of long and stretchy. But as you can see, they will be now becoming a minority, because anything which has been amplified will be now a sequence amongst the first and the second primer. Okay? And the more we do this, the more we only have these short sequences which have been amplified, and whatever was the original sequence is basically gone. This is diminished. And then we have our PCR product. And the key element is here the technical solution. Yeah? This is not to be compared with the replication, what you have in a mammalian cell, for example, or in a bacterial cell, because this requires much regulation, because every cell, even a bacteria, has to make sure it doesn't make too many defects. Yeah, you don't want to have too many mutations when you start from, single semen and single ovary, and then becoming billions of cells staying now in front of you. You have to make sure that the error rate is super low. This does not occur under these technical solutions because it would take just too much time and it's too complex and too costly. So what's the key element? Why a second primer is important? Well, if you would have only one primer, we would of course not be able to amplify as dramatically per cycle, and therefore have not the ability to achieve the signal to noise ratio over a very short time frame. Now, what is the key element here on the molecular level, and where does this 3' 5' comes from? If you think about how a nucleotide is being formed, let's assume this here is the so-called ribose. This is the sugar moiety every nucleotide has. And the denomination 1 to whatever usually is based on the carbon atom. Okay, so you have a carbon atom 1. This is the place where the nucleotide is sitting on it. And as you can see, it's ATCG in the case of DNA, or it's AUCG. So there's a difference between RNA and DNA, as you know it from school. And since we have the 2' side, the 2' side also differs between RNA and DNA, because in the DNA world, we lack the hydroxy group here. That makes DNA stable. This is the key element why DNA is stable. As compared to RNA, it has on the second and the 2' position a hydroxy group, makes it unstable. Basically, it's very easily digested by the hostile chemical environment in the cell. And that usually is something you would like to have, because RNA is in our cellular environment just a messenger, right? I just give you a little bit of information, but the information should also get lost after it has been transmitted. Then we get to the 3' position, which is now important for our discussion of 3' to 5'. The 3' position is one of the ends where basically the additional nucleotides are being added. Yeah, and so you're moving from the 3' end to the 5' end. 4' is not important, but then again, the 5', on the 5' end, we have now usually the phosphate group sitting, and that's exactly what you see now here, 3' end, 5' end. We are growing towards this end, so this end is continuously extending, and it is not possible for polymerases to do it the other way around. Okay, so this is a directed kind of a mechanism. Okay, as I said, this is not stable, and the actual DNA polymerase requires DNA. This is the type of enzyme we would like to work on. There are also other enzymes which can, transfer RNA to DNA information, but this is a very exotic type of an enzyme. Therefore, we have to get around the RNA going to the DNA world. So if we have an RNA virus, it's important to get to the DNA, and only then we can do the amplification and determine what type of infection you have. Blah, 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 blah. I think that's it. Good. So now what about the healing cycle? And again, keep in mind, we have one bucket. Everything is just put into it. And then we want to control when this reaction occurs, right? And how can we do this? What is the active element in this bucket? This is just a physical parameter, but the active element. The enzyme. So the PCR reaction has been optimized around the highest processivity temperature of the enzyme. Processivity means this is the average time an enzyme sits on a strand and just adds one nucleotide after the other until it falls off. Okay, there are polymerases who have a high processivity, stay there forever, and some of those guys just fall off very quickly. And you want to look for a technical solution for something which is a high processivity, obviously, right? Must not necessarily be our polymerases, but maybe from other organisms. And that's exactly what the people have been sneaking around. So which of these enzymes give me speed and high processivity? And the other thing is, I want to use now with temperature when something is being amplified, because I want to have a technical solution. I want to be in control of the reaction. Okay. And what the people then have been developing is this kind of a cycle. So I have a cycle where I start to almost cook my soup. To do what? To do the denaturation stage, going from a double strand to a single strand. I need to heat up because I have a very long stretch of paired nucleotides. You know, for hydrogen bonding is not really super duper strong, but you know, in a big environment, in a big ensemble of other hydrogen bonding nucleotides, actually you really have to cook it up to make it loose, and that they are not randomly, you know, binding a little bit on top of each other. It has to be really completely separated. And while we cool down, we actually do the hybridization step. So before those guys can start to find each other again, we have the primers in the soup. And the primers are, what are they? They are small, they're diffusing much faster. And while they're diffusing much faster around, you know, the big and heavy guys, they're just leaning a little bit relaxed in the hot water. And the primers have the time to bind. I mean, this is the physical picture, literally, okay? So when you do so, we have now all the starting conditions. And then there's a third step. And the third step is usually you go up a little bit in temperature. And you do so because you want to have here the highest processivity of your enzyme to do the replication step. Okay? Now, what is a key requirement for this enzyme in that soup here? If you think about this temperature regime. Yes. What is the highest temperature? Exactly. Yes, and this is something you don't want to do, right? So can we use our enzymes for that? Nope. Usually when we cook, we are well done, but we're not working anymore. So how the hell could these people find the right enzyme in those days? And this is this sneaky other storytelling. So there was one professor, he was walking through that American National Park with the geysers and all of this, Yellowstone, right? And then he looked into one of these hot pots and he saw that the hot pot was very colorful, green, red, and anything like this. And they said, what the hell can grow in almost cooking conditions? So those were bacteria. And they looked then into this bacterial enzyme called a bacterial polymerase, and that covered this regime. And the only thing what they did afterwards was to basically optimize and do a little bit of a protein engineering and so on and so forth to have today's technological solution. The technological solution looks like this: highest processivity, withstanding high temperature regimes, no denaturation. It can withstand all this, you know, technical conditions to just amplify whatever is there with a minimum amount of error. That's it. And then there are also other things, like when an enzyme starts to grow, you know, there's a chance that maybe there is a double strand bond in front of it, just by chance. So you're also even looking for an enzyme which has other functions, like it has also exonuclease activity. So it can eat away everything which is in front of it. And that's also a key element. Please.
But I just remember that usually the ribosomes do the translation.
Yeah, translation.
But I remember that in bacteria, they are different in size than in human. They have like 90S.
Yeah, a little bit different in size, but the reaction is the same, basically. Yeah. Just the regulatory, so the higher the organism, with respect to complexity, the more regulatory elements you have. A bacteria is actually not optimized for, I don't want to have any mistakes during my processes. Bacteria are so successful because they allow a certain amount of errors, because this gives them evolutionary fitness, right? That's a major difference between, you know, the time constant where bacteria is growing as compared to the mammalian cells, where it requires usually for you on average 25 to 30 years to replicate, right? So it's just a very different time scale. Okay, so voila. And now we have it. Now we can start talking about which type of PCRs have been actually developed in the past. And what I would like to do now is to talk about traditional PCR, and then I will talk about real-time PCR, because those are the two dominating forms. There are other, like digital PCR and so on and so forth. And when we have time, we're also going to discuss this. But for the moment, only that is important. From, again, the bird's view, if you think about amplification, on the y-axis, this would be the log scale on the copy numbers we are producing by cycling, ideally in such a way that every cycle we have 2 times more, right, over the cycling numbers. And as you can see, these are experimental curves, and they are not starting after the cycle 1, but they're starting to give you something at cycle, whatever, 17, 18. Okay. And then we have this exponential phase, which means that's exactly where we're duplicating the number of amplicons or copy numbers. And then we get into a linear regime, and then it saturates off. Now, my question for you is, why and why? And just think about the physical picture. Let's start with this. This is easier. Why is this not continuously going like this? Enzymes saturate with respect to what?
DNA.
Yeah.
Make the cycles go up, and they have the saturation of 28.
So that they have not enough enzymes to cover all the amplicons during the replication phase. Is this what you mean? Would be true, yeah. If you have too little number of enzymes, then of course you cannot uphold with the increasing number of what you have here. Sure. Yes, because what is the basic component? It's the individual nucleotides. You run out of fuel, ain't gonna work anymore, right? It will stop at some point in time. Other reasons. It is not a deterministic machinery. Well, the enzymes will die. They will also take damage. Or you have other errors in it. So at some point in time, your system will go into a plateau phase. Okay, that's about it. You're running out of primer, right? Exactly. But why that? I mean, I start to replicate after cycle 1.
Or just some chance of that.
Yes, exactly. And the technical issue what we're having. Exactly. The signal is just too low. So it's a little bit, of course, the biophysics behind it, but at the end of the day, it's also the sensitivity what we're having. I mean, we have single molecule sensitivity. It's not a big deal, but for a regular test, it usually looks like this. You take a few cycle times until there is a threshold reached. And if you think about this curve here, it's exponential. You could write down here voltage, and you can write down here current. And how does this look like now? It's like a transconductance curve, right? You have a threshold voltage where your field effect transistor turns on, and you have the saturation regime. This is your on state, right? And what you would like to have is a max between on and off state. In our case, it would mean somewhat specificity. It's a very quasi correlation, but it is at least a little bit of a comparison to your engineering world. Okay, so how do you do this now in reality? You have your bucket and you have nothing else but a heating element. And the only thing what you have to do is heat up, cool down, heat up, cool down. Hasn't changed for the past 70 years or so, just with the additional reagents being now faster. And that's basically a protocol as it would be for most of the PCR tests. And the only thing what you have to have now is one condition. You actually need to know what you want to amplify. I mean, you can just generate random primers, then you will also randomly amplify anything you would like to have. But if you want to have a clinically relevant test, you actually need to know the sequence of your SARS-CoV-2 virus, because you need to determine the primers. Primer design is a key element. This is the design area, you know. The enzyme will always be the same, the nucleotides will always be the same, but the primer design determines what kind of a test this will become. Is it now an influenza test? Is it now a SARS-CoV-2 test? You need to know which primer you have to have. And it needs to be specific for SARS-CoV-2 only. Otherwise, I might have a chance and I would amplify my influenza virus next to it. So those things have to be optimized by a known sequence. And what is Sanger sequencing doing? Well, Sanger sequencing basically does this a little bit differently. You start with not knowing what sequence you're having, and then you have to kind of dissect what you have been amplifying. And that's a different notion to PCR, where I need to know, at least for the primers, to make a test. And for Sanger sequencing, I need to know after I did my testing what I have been actually adding, nucleotide after the nucleotide, onto my sequence, which is unknown. Okay. And how do I do this? Usually when I'm working in this traditional plateau phase, I'm just amplifying until the reaction basically runs out of fuel. I have these gels. And these gels are nothing else but literally a gel out of a polymer, agar gel. And you have here pockets in the gel, and you put your samples inside. Usually you see this little bit of a bluish color. You know, DNA has no contrast, so you're adding something which is intercalating with the double strands. Those are colored molecules which give you a high absorption. And then you apply a potential between an upper and a lower electrode. And what the DNA is now starting to do, it starts to diffuse through your polymer network. And if the polymer network is basically the filter, you know, what is being filtered easily? Small objects. So that means under conditions of field strength like a few volts per centimeter, the small objects will diffuse pretty quickly to the counter electrode. And the larger, bulkier ones, they will take some time because they're always bumping against my biopolymer, which ends up to what? These kind of pictures. So if this would be my starting point, you know, I see that the largest objects will stay the closest to my starting point, and the smallest objects, like 100 base pair, has been moving quite distant. How do I know where I am and what size regimes are we talking about? Well, usually you're adding a calibrator with known lengths of base pairs, so that I have a so-called ladder, and the ladder indicates, you know, what size regime are we talking here about. Now, sorry. And now when you run your samples, you know, you can look, for example, and this would be old school. I know that I want to have now an influenza virus, and the piece of information which has been amplified should be in a size regime of 200 base pairs. If this band is there, it's good. It's not there, not good. No, it's very good, because then you don't have the infectious disease. Okay, so this would be the way how they did it in the early days. Okay, so how is this electrophoretic phenomenon actually working? Let's assume this bead here would be a piece of DNA, and any charged object, you know, remember we're working under physiological conditions, blah, 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 everything is charged. You will always have in your buffered solution where all of this is being in solution, you will have a cloud of counter ions. And the counter ions will mask, of course, my charges, right? But there is a so-called slipping plane. The slipping plane means this is the area where my counter ions, who are completely fixed to compensate for the negative charge of the DNA, as compared to these rather mobile ions where, you know, things become a little more fluid and ions are exchanging. And that means you usually have a net charge, which can then be applied to move your object through an electrical field you're applying to the system. And this is then called electrophoresis. Okay. This electrophoretic potential, what we're looking here, this is the so-called zeta potential. This is basically depending on the slip plane, the buffer conditions, the ion concentrations, what type of ions, and you name it, you have it. It's a non-intuitive value. That is basically the key element. Now, the beauty of having DNA is that every piece of that DNA stretch carries the same charge. Meaning, no matter how long that thing is, the mobility will be always the same. The only difference is if I add some barriers in between, like my gel, you know, the big guys take more time, and the small guys will just being mobile enough to walk through your polymer. Okay, do we also see this? You see here a little bit of velocity in millimeters per hour, versus the length scale in base pairs. And those are experimentally derived curves at a field strength of 2.5 volts per centimeter. Question for you is, where would you have the highest separation power? Any idea? Exactly, where the gradient is the highest. Yeah. So that is about the regime what you would like to have. And that's exactly how you actually then also tune your filter. Because your filter needs to be also adapted to your testing question, what you're having. Are you looking for some big bastards or for some small bastards? You basically have to tune also the filter. And usually, you know, you are somewhat in the regime of a few hundred base pairs to maybe a few thousands. That is where the polymer gels are usually being developed for. Okay, now we know how this all works. But I also told you that this picture, this schematic picture, what I've shown you before, is actually false. This is not true. It's not deterministic. Why? Because we're working with actually weak chemical bonds, hydrogen bonds. And we're working not under 5 Kelvin, but we're working under room temperature conditions. So that means if this would be a stretch of DNA, and this is now, you know, another piece of DNA forming this double helix. Yes, you will have a nice, usually a very nice and tight binding in the center, but not at the ends. The ends will be actually free floating away. Why? Simply because of thermal energy. The hydrogen bonds are not that strong. So that means the schematic picture that everything binds perfectly is not true. And I would like to show you that in an experiment, people have shown this. Or if you think about Gibbs-free energy always tells you when something is negative, you know, binds, positive, no bind. Okay, keep this in mind. And those are exactly the terminal base pairs what you're having. So, you know, this is hanging around. But still, for a primer, for example, where the last nucleotide is maybe not so perfectly binding, the enzymes seem to still being able to recognize this and start working on it. But again, you know, it's not a 0-1 situation. This is biology what you're looking at. Nothing is absolutely perfect, particularly when it comes to binding. So how did they derive this table? They used optical tweezers, 2 beads. On both beads, there was a double strand stretch, and then there's a single DNA stretch in between. An optical tweezer is a very tightly focused laser spot, and you have a pico-Newton resolution, and you can pull on these beads, pull them apart. And what they now did is they tried to check for the elasticity of this middle section here without a double strand pairing pair, or with a double strand pairing element here in between, which they checked with some fluorescent signal when this was there. And with this, they could plot basically the force curves, the elasticity, and calculate basically the energy values as they are plotted here in this table. Details are not important, just to give you an idea. Okay, this was traditional PCR. Yeah. We have a few minutes left. Real-time PCR. Real-time PCR, as you already get the idea, wants to quantify wants to give you in real time the amount of amplified sequences where in this exponential phase. And I literally, crossed this line here because there is a threshold value. And what you do is you want to save time. Yeah, you don't want to waste too much time by looking into this phase here. What you're actually looking at is you're looking into a situation where your signal is just giving you sufficient signal power to derive now something has been amplified in the exponential phase. It's like the threshold voltage in our field effect transistor. That is relatively nicely to determine. And that's the same thing what these people are doing when they talk about real-time PCR, because here you can quantify things as much as possible because you are assuming 100% reaction efficiency. Anything else stays the same, but the reporting is not different. Instead of working in a gel, we have to have now a different element because the gel only gives us ensemble information when we separate it out, right? But here we literally want to even work on a single molecule level and getting information out of it, which we can extract in the form of, for example, fluorescence light. And this is exactly one of the few cases where fluorescence detection is really strong. Basically, all of molecular diagnostics works with fluorescence detection, basically or everything. Whereas the central laboratory barely has any fluorescence detection there. This is all chemiluminescence and other elements. So, but how do you actually know what has been amplified, right? This is the big question. And for this, the people used the so-called first resonance energy transfer phenomenon to do the following. As I told you, when the people were looking for the smart heat-seeking enzyme, which shows for high processivity to just amplify, they also wanted to have an additional feature. That is that when this is the polymerase, this is the primer, the polymerase is walking this way, it eats away anything which is in its way. This is the exonuclease activity, okay? And you have this, then you have a feature. And this feature allows you now to look into what has been amplified. Because what you can form is, you can form this probe here. This is now the infamous Tachman probe developed by Roche after they bought the patent from Kerry so that he could serve much more. They developed now a probe which is sitting now on a sequence stretch in between your first and somewhere your second primer. Okay, this was the trick what they were doing. But still, fluorescence is there, fluorescence is there. So how can you discriminate fluorescence from sitting on a piece of DNA as compared to fluorescence because the probe is swimming around? You need to have some discrimination potential. That's exactly where the first resonance energy transfer comes in. And the trick is here that you actually have two fluorophores sitting in very close distance to each other. The first resonance energy transfer works with an efficiency which drops with the sixth power of the distance in between the two fluorophores, meaning a half a nanometer and there's barely anything left. And that's beautiful because this is exactly the situation you would like to have, because you would like to see only fluorescence when those guys are in a very defined close distance to each other, as compared if those two fluorophores would be separated from each other in a liquid, they will have on average a larger distance to each other, so you will not see a fluorescence. So what fluorescence are we talking about? The concept is that you have two fluorophores with an overlapping area between the emission of fluorophore 1 to the excitation area of fluorophore 2. So this is the wavelength, right? You have in the bluish regime, you excite the first fluorophore. This dude emits in the green, but the green light is being reabsorbed in the same second because they are very close in distance to each other, and basically fills up your energy levels to emit at the end of the day red fluorescence. And that's the thing you're detecting. If this guy is there, you know that those probes don't exist anymore. Make sense? Because there's no transfer. Or you detect the emission. The emission will be there and high, when this distance is not given anymore because there's no energy transfer. So those are the modalities how you can play with the Fred effect. Make sense? Yeah? Okay. So, and how does the sequence now looks like? This is the double strand starting material. You have your primers, you have your probe, you have the polymerase, you have the nucleotide triphosphates, everything put into one bucket again. You mix it and you just do your heating cycles again, meaning if you're working with your heating cycles, also your probe needs to be adapted to the primer design, to the conditions, how you're measuring, and so on and so forth. So PCR looks simple, but at the end of the day, it's actually also complex to optimize the conditions for achieving your goal of a clinically relevant assay. So how does it look like? In the second, you had your heating step to denaturate the double-strand DNA. Primers will bind, but also the probe will bind in between the stretch of primer 1 towards 2. And during the polymerization phase or the elongation phase, the enzyme will show its exonuclease activity. It will digest anything in front of it. The donor and the acceptor or the quencher, however you want to call it, they will be separated from each other. And that means those fluorophores will be completely separated. And you have then either high intensity or low intensity, whatever you're looking at. Okay, this is now the specificity you're getting. And that allows you to tell how much I have been amplifying. This is real-time analysis. You can even do it on single molecule level. Okay, but you're working in a clinical setting, meaning you need to make sure that whatever you are measuring, and as you have seen, the starting conditions are not perfect. The starting conditions may vary. If you have a high titer of virus in your body, you know, super high HIV infection, or you have a super low HIV infection. But you need to have some kind of a calibration to understand how many copies you actually made, right? So how do you do this? Well, the PCR test you have been developing for HIV was based on, I know my sequence, okay? When I know my sequence, I can also fabricate now a calibrator. Basically, I make fake viruses in defined concentrations. And what I do now is when I do my test for each and every test conditions, just to make sure that everything is completely comparable, I'm running also my calibration material. My calibration material means I have basically my fake HIV virus from 10 to 0 copies to 10 to 7 copies. And I also do my same PCR reaction with the probe on it at the same time as I'm measuring my sample. When I do so, this is the relative fluorescence on the y-axis. So this is the signal I'm taking out from the Fred transfer or the loss of Fred transfer versus my cycle numbers. And in the second, where I have a threshold, and the threshold is usually 10 times the noise level, this gives me a safe fluorescence signal, which I can pick up to quantify in the exponential phase. This gives me a certain cycle number. And the less starting material I have, what does this? Okay, the less starting material I have, the more cycle times I need or cycles I need to achieve my threshold value. Okay, makes sense. So if I do this with my calibration material, I can plot my threshold cycles against my starting quantity, and I generate a calibration line, right? And then I'm just looking for my cycle time of the unknown sample I had. Maybe this was this value here. I just plot it to the cycles, and then I know what was the starting quantity of HIV viruses per milliliter or microliter of blood or anything like this. That's the way it's done, okay? And now you can treat an HIV patient because I see you have a lot or you have less, but you don't know yet what kind of a sequence this HIV virus has. For this, we need to have sequencing, okay? I only know which type of a virus you're having, which is, of course, another relevant element for doing the therapy. What's the beauty of this concept? I don't need the gels anymore. I'm just, literally looking for fluorescence intensity and I just have to pick up the fluorescence intensity versus time. Now I show you, oh, hang on. Do we have here a video to share? Yes. I'll show you the light cycle. Because whatever you start in here, it could also block your PCR reaction. For example, hemoglobin is a killer for PCR. You have to get rid of the red blood cells and the hemoglobin before you start to do PCR. Many surfactants are a killer for PCR because they kill the enzyme, and so on and so forth. So you need to look for the right composition of your patient sample so that you can really do whatever you would like to do. So, and then the first product on the market, which was really a success from perspective of, I don't want a pipette 10 times, but I want to make sure that I have an integrated workflow. And I can even do point of care testing, because infectious disease diagnostics usually something you would like to do at the acute care. And that was the Cepheid workflow. And I show you this because by chance I know the founder of this company, and he told me all the dirty details of how he became a successful or not so successful company. Let's see how it looks like. So what you have now in your hands, or what is being passing through these little small boxes, this is basically all the working steps, what you have not even seen the schematics yet. From, sample comes in, then you have to do the sample preparation, then the PCR is being done, and then you do the analysis. By real-time PCR, it all happens basically magically and at once in a black box. This is what we call point-of-care diagnostics. And the system basically works like this. You have to wash, you have to extract DNA or RNA, you have to purify it, and you add the so-called master mix. When people talk about the master mix, this is the mix of reagents and enzymes which you have to put in so that actually your reaction occurs. Then you have the amplification stage, the detection, and it's the analysis part. So this will be the analytics. And all of that happens basically in one pocket. It's kind of a walk-away testing. You just have to press the button and that's it. So Kurt was the founder of the company. He's one of the gods in MEMS, actually, but he spent his money also into this company because he believed in this. And they went bankrupt almost 10 times or so because it was super difficult to integrate this workflow. Why? Because what you put in could be super, super variable. I mean, one patient to the other, that's the difficulty what we're having. Technology always works, but does the technology work for all the patient samples? That's the tricky part. And then you have to prove that this is also working. So in either case, they came up with this kind of a blue box. What you're having, you can also open the lid if you want, or play around with it, and you see all kinds of different compartments. And they have basically a mix of different pistons, dried reagents, where you resolubilize, where you basically bring the different chemical compounds into reaction with each other. And the difficulty the company had was the molding and the assembly of those cartridges was actually pretty expensive. You had 12 components. You needed to mold 9 of those components. You had different kinds of reagents. The complexity was very, very high, which made the costs for this test also very high. And this was, of course, a limitation for the community to take this type of thing over. But then they had the first Christmas present, and the first Christmas present was this letter to Senator Daschle, and it involved anthrax. So those were these domestic terrorist attacks in the US. And what that led to is that the US post offices screamed up and said, "Oh, we have white powder here. I don't know what this is. Is it cocaine, or is it sugar, or is it anthrax? I need to have a test for that." Okay, most of the cases was cocaine, most likely, but in some of those it was anthrax. So now they started to sell. Yeah, it's not IVD testing, it's just sell something to the US post office. But a US post person, is not an expert in vitro diagnostics. He wanted to have what? A walk-away solution. This was the starting point. Okay, and with this, they were lucky enough, and then finally had now final design, final access to the market. They had some income, and that knows what you see here at this very edge. This is actually where the fluorescence detection occurs. And let's have a quick look here on that video so that you see a little bit the difference to the 96 well plate, the standard format for doing this kind of testing.